All right, everybody, we will go ahead and get started now. Thank you so much for joining us. Welcome to our Young Scientist webinar series. This is the first month and our first series that we are doing this. Kicking it off here with Caitlin Magel today, and she's going to talk about the interaction of climate change and human-driven changes in the Pacific Northwest estuaries and sharing perspectives from seagrass and macroalgae. And then our presentation next month will be on November 11th, and it will be under the cover of darkness, Tracking Oregon's Marbled Murrelets with Lindsay Adrian. And we are going to host this series the second Tuesday of every month through April of 2021. And just real quick before we get started in the presentation, I want to share a bit about the Cape Perpetual Collaborative. It came together and organized a few years ago to foster conservation and collaboration within local communities. And our three guiding principles are community engagement, leveraging resources, and engaging in partnerships. And as you can see here at the bottom, we have a variety of founding partners, including federal agencies, state agencies, nonprofits, and also the Confederated Tribes of Coos, Lower Umpqua, and Slough Indians. Our focus is on the Marine Reserve, Cape Perpetual Marine Reserve and protected areas. And I just always like to highlight this, just kind of give you a real quick visual of the Cape Perpetual Marine Reserve. It's Oregon's largest marine reserve. It spreads pretty much between Yachats and Florence with North Marine Protected Area and a Southern Marine Protected Area. And this goes out 3 nautical miles into the ocean. And the image on the right here is kind of a visual of what you would see underwater, which is mostly sand and gravel. But this really dark area is an isolated rocky reef that's very unique to the Cape Perpetual Marine Reserve. We do host a variety of community science opportunities around the Cape Perpetua area, as well as host a variety of different events. Many of these projects are during the summer or very seasonal, but we do host monthly beach cleanups at Cape Cove Beach. This year, we will continue to organize and coordinate the Fall-Winter Cape Perpetua Speaker Series as well. And that will be on Saturdays at 10 a.m., November through March, and that starts on November 7th. And our upcoming annual Cape Perpetua Land Sea Symposium is coming up on November 19th, and we are in the process of finalizing that agenda. I'm getting excited for this year's lineup, and it will be hosted in a virtual format, so stay tuned for more information on that. You can stay up to date with upcoming events and everything happening on our website at capeperpetualcollaborative.org, and just click on the events tab, and it will you can click through the calendar and kind of go to any of the events and learn more. And feel free to connect with us on our Facebook and YouTube as well. I will be posting this presentation within 24 hours, so by tomorrow evening, and so you'll be able to rewatch it or share it with others. And if you have any questions as Caitlin's going through her presentation this evening, go ahead and pop them into the Q&A box. You will see that if you hover over like where you can mute yourself, and just plug your question in there, and we will get to those at the end of tonight's presentation. And with that, I'd like to introduce Caitlin Magel. She is am I pronouncing it right, Magel?
It's Magel.
Magel, good. Caitlin Magel. She's a PhD student at Oregon State University, and she studies ecology, seagrass, and macroalgae in the Pacific Northwest estuaries, so she can better understand adaptation and biochemical changes in the environment. Her academic interests include estuarine ecology, land use, biogeochemical cycling, and ocean acidification. That's a lot of big words. And with that, Caitlin, you can take it over.
Thanks so much,
Yes.
Tara. Let's see. Okay. Yeah, thank you, Tara, for that introduction, and thanks to the Cape Perpetua Collective for organizing this webinar series and including me as a speaker. I'm excited to talk to you this evening about some of the research that I recently completed for my PhD at Oregon State University. And I'm especially excited because I get to talk to you about one of my favorite ecosystems, which is estuaries. Estuaries are highly dynamic ecosystems where freshwater rivers and the ocean meet. They're defined by transition from upland to ocean and from freshwater to saline, and they're defined by variation, spatially and temporally, both within and among estuaries. And over the last 10 years, including during my PhD and through several positions at the Hatfield Marine Science Center, I've had the privilege to do research in just about every estuary on the Oregon and Washington coast. I can tell you that no two estuaries are alike. And from my talk today, I hope you'll gain a better understanding of how and why estuaries of the Pacific Northwest coast differ, and why it matters in the context of future environmental change. Estuaries provide many important ecosystem services, which are the direct and indirect benefits that humans receive from natural ecosystems. For estuaries, these include services such as coastal protection, filtration of nutrients and pollutants, and maintenance of lucrative fisheries. But their location at the land-sea interface also makes them susceptible to many impacts, such as excess nutrients and toxics, overharvesting, and habitat loss. In addition, global change is causing other emerging stressors, such as warming temperatures and ocean acidification. Given the variation that we know exists within and among estuaries, it's reasonable to expect that they differ both in the services provided and in their response to these various threats. Vegetated estuary habitats, such as seagrass beds and salt marshes, provide particularly valuable ecosystem services. And so one of those critical habitats, the focus of my dissertation research, is seagrass beds. Seagrasses are rooted flowering plants that can inhabit shallow subtidal and intertidal zones of estuaries. In the intertidal zone, seagrass distribution is primarily limited by the amount of aerial exposure or desiccation stress as the tide goes out and comes back in. And then in the more subtidal zones, distribution of seagrass is limited by light availability as it penetrates through the water column. And these habitats provide habitat for many important fish species in the Pacific Northwest, like herring and Dungeness crab, as well as juvenile salmon and shorebirds. In the Pacific Northwest, our common species of seagrass is the native eelgrass, or Zostra marina. And in addition to eelgrass, the other common macrophyte, or kind of large plant species in Pacific Northwest estuaries, is macroalgae. And specifically, we commonly find green macroalgae in Pacific Northwest estuaries, which include species from the genera Ulva and Enteromorpha. And this picture shows kind of a raft of green macroalgae in Uquina Bay that settled out on the eelgrass, on top of the eelgrass bed during low tide. The reason that I study seagrass beds is partly because historically there have been dramatic declines in seagrass habitats worldwide. We think that 30% of seagrass beds have been lost worldwide, and that declines continue to accelerate. So given their importance globally and in the Pacific Northwest, we need to better understand the environmental factors that determine the success of seagrass ecosystems. One of these factors that you might be familiar with is upwelling. The Pacific Northwest coast of the US is influenced by the Northern California current, which experiences seasonal upwelling of cold, nutrient-rich, and low pH waters to the coast. Upwelling is caused in our region by winds that blow equatorward or north to south along the coast. Specifically in summer, we see these conditions. And in combination with the Coriolis effect, these winds drive surface currents of the ocean offshore. This movement of surface currents causes the upwelling of deep water at the coastal margin. This water's cold, like I said, nutrient-rich, and low pH. And because of this kind of built-in refrigerator of cold water, it's presumed that upwelling zones may be more robust to global warming, although this assumption has yet to be adequately tested. In addition, along the US West Coast, upwelling strength and the duration of the upwelling season increases from north to south. So these spatial gradients in upwelling, combined with things like coastal topography and watershed drivers, create gradients, a latitudinal gradient, in nutrients and temperature among Pacific Northwest estuaries, where northern estuaries are generally warmer and lower in nutrients, places like Willapa Bay and Netarts Bay, and southern estuaries are colder and typically higher in nutrients, like Coos Bay and Yaquina Bay. These factors then drive opposing latitudinal patterns in seagrass and macroalgae, where we have tend to have higher eelgrass biomass in northern estuaries compared to southern estuaries, and higher macroalgae biomass in southern estuaries compared to northern estuaries. Therefore, the dominance of macroalgae in southern estuaries is associated with high nutrients and cold water, whereas seagrass production, on the other hand, is negatively correlated with temperature and nutrients. So previous research has found that northern estuaries tend to be eelgrass dominated with very little green macroalgae, and southern estuaries tend to be macroalgae dominated with relatively less eelgrass biomass. But we know that there's significant global and ocean change that's happening. Carbon dioxide emissions are resulting in complex global changes with multiple impacts to oceans and coastal communities. As this infographic shows, the ocean is absorbing heat and carbon dioxide, and this has resulted in an ocean that's more acidified, warmer, and less oxygenated. And these changes threaten many species, ecosystem functions, and important services. And taken together, the effects of global change are creating scenarios that are environmentally, ecologically, and societally novel. Scenarios that are increasingly difficult to anticipate and to manage for. Therefore, the research for my dissertation aimed to answer important questions about the drivers and consequences of global change for coastal estuarine habitats in order to address important conservation and management challenges in the face of global change. Building on these novel scenarios, we've also seen a global increase in the occurrence of marine heatwave events. Marine heatwaves are defined as prolonged periods of anomalously high sea surface temperature, and they represent an acute thermal stress that differs from the gradual warming that oceans are also experiencing from global change. Here are a number of high-profile marine heatwave events that have occurred in the last 2 decades. The one that should stand out the most and that you're all probably most familiar with if you're tuning in from the West Coast was the 2013 to 2016 Northeast Pacific Ocean marine heatwave. This is the largest marine heatwave on record in both spatial extent and temporal duration. And although the marine heatwave formed offshore, sea surface temperature anomalies were observed along the US West Coast, starting in January of 2014 and extending through the summer of 2016. These events are predicted to increase in both frequency and magnitude in the Pacific Ocean with climate change. So it's important to understand the effects that these marine heat waves have on coastal ecosystems and species. The documented impacts that have been shown thus far from the recent marine heat wave were geographical shifts in species ranging from tiny copepods to large sharks, mass strandings of marine mammals and seabirds, and closures of economically important fisheries like the Dungeness crab fishery. In addition, offshore foundational macrophytes, including giant kelp and bull kelp, were also strongly affected. But what about estuaries and estuarine macrophytes, including seagrass and macroalgae? Marine heat waves have been implicated in seagrass declines elsewhere in the world, but there's no information regarding how the Northeast Pacific Ocean marine heat wave event was felt by estuaries along the Pacific Northwest coast and how estuarine macrophytes were impacted. So for part one of my talk this evening, we're going to look at how the marine heat wave affected eelgrass and macroalgae across 4 Pacific Northwest estuaries. And then in part two, we're going to look at whether macrophyte responses and drivers were consistent along the estuarine gradient from marine dominated to river dominated, and to see whether those responses and drivers change along that gradient. So part one, how did the marine heat wave affect eelgrass and macroalgae across 4 estuaries? So I'm going to represent my hypothesis based on the latitudinal differences that we already discussed about Pacific Northwest estuaries. And I'm going to represent this on a die plot with 2 axes. So along the vertical axis is estuary temperature from estuaries in the south that we know are generally colder to estuaries in the north that we know are generally warmer. And then along the horizontal axis is the kind of relative macrophyte abundance and the patterns that have historically been observed in macrophytes, from macroalgae dominated estuaries on the left hand to eelgrass dominated estuaries on the right hand. And we can place estuaries where they've been historically observed. So Coos and Yaquina being generally colder estuaries that are macroalgae dominated, to Netarts and Willapa that are generally warmer estuaries and are eelgrass dominated. So I'm going to represent my hypotheses about how these estuaries will respond. And if macroalgae prefer colder temperatures and eelgrass prefer warmer temperatures, then Coos and Yaquina, which tend to be colder, could actually move from being eelgrass dominated or macroalgae dominated to being eelgrass dominated as a result of the marine heat wave. And then the response of estuaries like Netarts and Willapa could take 2 possible routes. The first is that perhaps the previously kind of lush eelgrass beds and the warm waters of Willapa and Netarts provide a refuge for eelgrass to withstand disturbance, and that eelgrass would respond favorably to the marine heat wave, moving right in this die plot. Or conversely, eelgrass may be already near their thermal boundary, such that the additional thermal stress of the marine heat wave would actually cause eelgrass to decline in those estuaries. So the way that I was able to test these hypotheses was using the 4 estuaries that you see here. I had summer macrophyte surveys from marine dominated sites in each estuary over 3 time periods. So my before marine heat wave time period was from 2006 to 2010, during the marine heat wave from 2015 to 2016, and then after the marine heat wave from 2017 to 2019. And so for all these time periods, we characterized eelgrass and macroalgae biomass at each of these sites in the 4 estuaries. In addition, I compiled temperature records from each estuary, going back in time as far as I could find data available. I also did this, looked at the temperature data for an offshore buoy located near Cape Blanco, Oregon. And so this is showing the propagation of the marine heat wave into kind of the near shore region in Southern Oregon. And I'll orient you to this figure because then I have 4 that look just like it for the 4 different estuaries. So here we're showing estuary temperature in Celsius from 5 to 20 degrees C from July of 2013 through July of 2016. Here, the black line is showing the long-term climatology or the long-term kind of seasonal mean temperature at this site. The green line characterizes a threshold, so above which would be considered a marine heat wave, and below which would not be considered a heat wave. And then the actual observed temperature for each date is the thin gray line. And you can see anywhere that thin gray line passes above the green threshold, it's infilled with red, indicating a marine heat wave event. So you can see that over the time period of the marine heat wave, it wasn't a consistent elevated temperature offshore here, located in Oregon, but we saw kind of punctuated time periods of these marine heat wave events, some of which lasted for months or even 6 months at a time over the kind of larger time period of the marine heat wave. So now I have 4 of the same plots showing you the estuarine temperatures from Willapa Bay in southern Washington, and then Netarts, Yaquina, and Coos Bay in Oregon. Again, showing that same time period from July of 2013 through July of 2016 for each estuary. And what we see is that these estuaries experienced heat waves coincident with the marine heat wave event offshore, showing that those warm temperature waters were propagating into the estuaries and being experienced there. But there's also differences in the estuaries, kind of underlying differences in their temperature, in their seasonality, in their temperature. And we see, like I told you earlier, that especially in summer, estuaries like Willapa and Netarts tend to have relatively high summer temperatures, and that's shown in the long-term climatology. And then the marine heat wave event is increasing temperatures above that. Whereas Yaquina and Coos tend to have kind of much more stable temperatures across season, shown in this long-term climatology, the black line. But we're still seeing those marine heat waves punctuated throughout the time period. So what effect did these temperature changes have? So like I said before, we characterized biomass before, during, and after the marine heat wave. And I have 3 plots that I'm going to show you: eelgrass biomass, macroalgae biomass, and then total combined biomass. So this first plot here is showing eelgrass biomass before in white, during in gray, and then after the marine heat wave in dark gray for the 4 estuaries. So Willapa on the left to Coos Bay on the right. And what we see in those 2 warmer estuaries, Willapa Bay and Netarts Bay, is that eelgrass biomass consistently declined before, during, and after the marine heat wave. Whereas in Yaquina Bay, eelgrass biomass was relatively consistent over those 3 time periods, shown by these box plots that are relatively aligned. And then in Coos Bay, we see something even different, which is that eelgrass biomass increased significantly during the marine heat wave event, and then decreased back to kind of a similar level that was seen prior to the marine heat wave. Overall, however, eelgrass biomass was highest before and during the marine heat wave event across all the estuaries compared to after, showing that in general, eelgrass biomass declined through this marine heat wave event, kind of across the coast. So now the same figure here, except we're showing macroalgae biomass for Willapa, Netarts, Yaquina, and Coos. It's a little hard to see because in general, macroalgae biomass is very low in Willapa and Netarts, but in general, macroalgae biomass was lowest during the marine heat wave compared to before or after in those two estuaries. And the same was true in Yaquina, a very dramatic decline in macroalgae biomass during the marine heat wave compared to before or after. And then kind of an overall decline in macroalgae biomass during and also after the marine heat wave event in Coos. And then kind of to bring it all home, if we add the two together, so this is total biomass, including seagrass and macroalgae. In general, we see pretty dramatic declines in estuarine macrophyte biomass. These are showing, especially in Willapa and Netarts, a greater than 50% reduction in biomass on a per area basis, showing that there are really dramatic changes that have been observed over this time period, kind of aligning with the marine heat wave and responding in a way that is similar to what we would predict. And overall, biomass of macrophytes across the coast was highest before and lowest after the marine heat wave. So if we return to my hypothesized responses, these are the directions that I thought the estuaries might move based on their dominant macrophyte and also their general kind of estuary temperature. But what we saw is that in general, macrophyte communities at these marine dominated sites were not static. That's not surprising given the responses that we saw in offshore kelp species. So showing that macrophytes in estuaries are also sensitive to the acute thermal impacts of the marine heat wave event. But the changes in biomass varied between estuaries, and they appear to be driven by the underlying differences in temperature and dominant macrophyte type. So what we saw is that Coos Bay went from being macroalgae dominated all the way over into being eelgrass dominated because of large declines in macroalgae biomass and then subsequent increases in eelgrass biomass. Yaquina Bay didn't change much, although kind of overall macrophyte biomass declined. And then Netarts and Willapa responded in kind of our second hypothesized way, which is that eelgrass was clearly close to its thermal limit. And so the additional thermal stress caused by the marine heat wave caused eelgrass biomass to decrease significantly. So if we think about kind of what this could mean for the future, rising temperature from global change and also more frequent marine heat wave events are likely to disproportionately negatively impact macroalgae. However, eelgrass may be positively or negatively affected depending on if estuarine water temperatures more frequently exceed tolerance thresholds. The marine heat wave affected Pacific Northwest estuaries in different but predictable ways. In estuaries that were already warm and likely close to their thermal limit, eelgrass biomass decreased significantly. In estuaries that were generally cold, the additional warming caused by the marine heat wave may actually favor eelgrass biomass up to a point. And in general, it appears that eelgrass in the Pacific Northwest may be locally adapted to cooler temperatures compared to elsewhere in their nearly global distribution, including much of the east coast of the US. Okay, so for part two, I looked at whether the macrophyte responses and drivers were consistent across locations within a single estuary. So for this, I focused on Coos Bay, investigating the environmental controls and associated macrophyte responses across sites. And although I just told you that eelgrass biomass actually increased in response to the marine heat wave event in Coos Bay, that actually wasn't the case at all locations in the bay. In particular, upper estuary sites in South Slough, those that were further from the marine dominated mouth, experienced dramatic declines in eelgrass over the last 20 years, and particularly over the last 4 to 5 years. These pictures were all taken at the same site, Volino Island in South Slough. And you can see that it goes from being a very lush, densely vegetated eelgrass bed to being sparsely vegetated. And much of the green that you see in this final picture in the upper right is actually macroalgae, and there's very little to no eelgrass left at this site. South Slough is the southern arm of the greater Coos Bay, and kind of fortuitously, a large portion of the estuary and its watershed are encompassed by the South Slough National Estuarine Research Reserve. So because this is a research reserve, there's been paired water quality and macrophyte monitoring at 3 sites along the estuarine gradient since the early 2000s, which provided a unique data set to investigate both the drivers and the consequences of variation in macrophyte abundance. So if we look at Barview, this is the site that I talked about in part 1. So this is the marine dominated site where we saw an increase in eelgrass biomass in response to the marine heat wave event during 2015 and 2016, compared to before and after, and an overall decline in macroalgae biomass, which is shown by the triangles. So what about these other locations? This is Volino Island, which is kind of a mid-estuary polyhaline site. And overall, we see, eelgrass biomass kind of bounces around over time and then generally starts to tank starting in 2014 without any apparent recovery. And then finally, Danger Point, which is the most river-dominated location, similarly has relatively consistent eelgrass leading up, in this case, until about 2016, and then it goes to 0 and basically stays there. And I started doing my PhD work in South Slough in 2016, which is right around the time that scientists at South Slough Reserve started noticing a thinning and general kind of decline of the eelgrass at that Volino Island site, which is where they survey most frequently. And I don't have time to tell you the specifics, but I did an analysis to identify kind of the timing of the eelgrass declines. And this declining biomass was really identified in winter of 2016. However, there was rising spatial variance in the eelgrass bed that actually preceded the declines, starting in late 2014, early 2015, which seems coincident with the timing of the estuarine heat waves that we observed in Coos Bay related to the marine heat wave event. But obviously this is all correlation, and so we needed to do a little bit more digging to see if temperature was actually responsible for the eelgrass declines that we observed at Volino Island and Danger Point in South Slough. And also to see if there were other interacting environmental factors that could have contributed to these patterns. So to answer these questions, I compiled environmental data sets across South Slough to describe what I'm calling 5 environmental regimes. So these were data related to temperature, nutrients, watershed factors, including watershed disturbance like forestry practices, Ocean-related factors describing upwelling and currents and other kind of regional ocean indices like PDO, which you might be familiar with. And then a regime describing ocean acidification and hypoxia-related factors, so pH and dissolved oxygen. So these represent all the possible drivers that I could think of that could be related to eelgrass and macroalgae dominance dynamics across sites. But there's too many variables here, variables that are related to one another positively and negatively, related to one another directly and indirectly, which prevented me from being able to just kind of throw them all into a simple analysis and come out with the answer. So instead what I did was to combine them into these radar plots by regime. So here are my 5 regimes that I just talked about that are basically describing how these environmental factors differed across space and over 15 years in South Slough. And by plotting them in this way, the variation in these factors across space and time is basically reduced to 2 axes. So we have dimension 1, which is the horizontal axis, and dimension 2, which is the vertical axis in each of these plots. And then we have the individual elements like precipitation or watershed disturbance or water turbidity that are either associated with one or the other or kind of partially associated with each axis. So we have 2 axes of 5 different regimes that are basically reduced into 10 possible explanatory variables with which I could try to describe eelgrass and biomass dynamics in South Slough. So I took all 10 dimensions of my 5 different regimes and performed model selection where I eliminated variables or individual axes of these different regimes that were not important in describing eelgrass and macroalgae biomass. And what was left after I did that process is basically the best model representing the spatial and temporal variation in eelgrass and macroalgae biomass in South Slough. So what I found for the best model of eelgrass biomass included dimension 1 of the temperature regime, dimension 1 of the watershed regime, and dimension 2 of the ocean acidification and hypoxia or OAH regime. So what this means is that I found that that especially mean water temperature and mean air temperature were negatively related to eelgrass biomass. In addition, watershed disturbance, metrics of watershed disturbance were negatively related to eelgrass biomass. And then finally, dissolved oxygen and pH, as well as photosynthetically active radiation, or basically the light available, was positively related to eelgrass biomass. And I found the same 3 variables to be important for macroalgae biomass. So again, macroalgae is negatively related to temperature, negatively related to watershed disturbance, and potentially also water clarity or turbidity, and then positively related to these metrics related to ocean acidification and hypoxia. So in general, macrophyte biomass for both macroalgae and eelgrass in South Slough was negatively related to temperature, kind of further including this impact of the marine heatwave event, as well as potentially a combined effect of that thermal heat stress with light limitation that could have been caused by watershed disturbance that kind of reduced water clarity and imposed some light limitations on the plants. And then I also found that biomass was positively related to pH and dissolved oxygen. And this indicates what another aspect of my dissertation was looking at the influence of macrophytes on water quality via the process of photosynthesis and respiration. So when these plants are submerged and photosynthesizing underwater, they're actually taking out carbon dioxide and releasing oxygen, which creates a drawdown of pH associated with that CO2 being taken out of the water, and then an increase in dissolved oxygen related to photosynthesis as well. Unfortunately, I don't have time to go into this more, but again, I looked a little bit closer at what this actually means for future environmental change related to ocean acidification and hypoxia. So maybe I'll give another seminar about that later. So overall, I think that my research really highlights several things. The first of which is the value of long-term data sets for understanding the effects of global change. This is particularly true in estuaries because of their highly dynamic and variable nature. For instance, it's only because of the 10 to 15 year data set that I was able to compile that allowed me to tease apart the effects of a multi-year marine heat wave event. And these marine heat waves are really just coming on our radar as a consequence of global change. In addition, I found that Pacific Northwest estuaries may be less robust to environmental change than was previously thought. I found that estuarine macrophytes, including seagrass and macroalgae, experienced dramatic shifts in less than 2 decades that were at least in part associated with increasing temperatures. And these changes to ecosystem function, especially related to the eelgrass declines that we observed in South Slough, represent a substantial change in ecosystem function and a loss of important biogeochemical processes and ecosystem services. And I feel that my research builds on and extends our understanding of coastal vulnerabilities, as well as estuarine ecosystem dynamics. And although we found that the drivers and consequences of global change may be estuary specific, even within Oregon and Washington, there's still predictability in the observed patterns. And these contexts provided by my dissertation research, a portion of which I've been able to share with you tonight, will allow us to be better prepared to address future coastal conservation and management challenges in the face of global change. So with that, I'd like to acknowledge several people and groups, in particular my collaborators, including my PhD co-advisors, Sally Hacker and Francis Chan at Oregon State, and then a number of people, but particularly Ali Helms and Adam DiMarzo at the South Slough National Estuarine Research Reserve, and then a former graduate student of my advisor, Sally, Margot Hessing-Lewis, whose work between 2006 and 2010 really kind of set the stage for a lot of the research that I was then able to build on. And then I received funding that supported this research from my home department at OSU, the Department of Integrative Biology, as well as a graduate fellowship from the National Estuarine Research Reserve system. And then a number of people provided data and wisdom that helped me be able to complete this work, including Burke Hales, T. Chris Mashon-Kalura, Andy Sherbier, Jen Haydock, and John Schaefer, who at the Confederated Tribes was instrumental to being able to collect some of the data that I collected there. And then NANUS, a lot of the temperature data going back to the late 1990s was publicly available on the NANUS website. And then finally, an army of Oregon State graduates, undergraduate students, as well as summer interns at the South Slough Research Reserve contributed to field and lab help for my project. And I'm grateful for the hours that they committed, mostly volunteered, to my science. and that they weren't deterred by crawling in the mud as the tide came in, which did happen on several occasions. And with that, thanks so much for your attention, and I welcome any questions.
Thank you so much, Caitlin. While we let questions roll in, I have kind of a two-part question.
Okay.
One is, oh, then I forgot. First, what was something that surprised you throughout your research, or did anything surprise you?
That's a great question. I would say the thing that surprised me most is that when I started my PhD in 2016, the reason that I chose to kind of focus my research around South Slough and Coos Bay was because of the lush, productive, extensive eelgrass systems that existed there. And right at that time is when they started to notice these declines. And so every time I went back to survey, there was less and less eelgrass. And that really kind of flipped my research on its head from looking at the benefits of these lush, productive ecosystems to like what happens when we lose it. And so I was, at first very disappointed about what was happening and dejected about the future of my research, but was able to answer some really actually exciting questions that I think were actually potentially even more informative than the questions that I would have been able to answer otherwise.
Very cool. It's really amazing how it's all connected, and sometimes you don't realize it until you start digging into things.
Absolutely, yeah.
And then what's been your favorite part of the research? Do you like going and crawling around in the muddy estuaries, or what's been your favorite part?
Yeah, I would say that first of all, like my favorite part has always been the field work, and that's kind of what drew me into this research in the first place. I came out to the Hatfield Marine Science Center as a summer intern more than 10 years ago and worked in the estuaries. I studied mud shrimp.
Mm-hmm.
And despite the fact that it's very dirty and sometimes challenging work, I just loved being outside, being able to see these natural ecosystems and kind of investigate different aspects of them. And then, more recently, as like my PhD really came together this summer, just the finding answers to the questions in this messy data that you think is never going to tell the story that you were hoping it would tell, or that you're never going to be able to kind of wade through the mess of field data. And then you do, and an answer arrives, and maybe it's the answer you were expecting, or maybe it's not, but you're able to find kind of clarity through that process. And that is something that I've only really come to appreciate in the last year as I've wrapped up my PhD.
Very cool. And then we have a question here. How might loss of plant biomass in estuaries affect decreasing ocean pH?
Yeah. So ocean acidification, which is related to kind of the global change and broader patterns of fossil fuel burning and CO2 emissions, is driving down pH in the oceans. And then we're seeing those kind of low pH events. And on the West Coast, we're kind of on the forefront of that because we already are impacted by ocean upwelling of low pH water. And so we're seeing those effects in the estuaries. You may be familiar with it impacting oyster hatcheries, eroding Dungeness crab shells. There's so much research coming out about that. And the plants have the ability to actually modulate or mitigate some of those effects because they release oxygen and take up CO2 during photosynthesis. And so there's this really strong feedback between those two. And when we see the declining plant biomass, there's less photosynthesis happening. And so there's less mitigation of those potentially harmful pH conditions. And so, you know, I don't think that the estuary plant biomass will necessarily affect ocean pH, but certainly the declining plant biomass, like we've observed in a number of estuaries, but particularly in South Slough, will have dramatic consequences for the OA mitigation potential of those estuarine habitats. And so could have dramatic consequences for things like juvenile Dungeness crab, which are sensitive to pH, and also juvenile salmon, which are sensitive to low dissolved oxygen concentrations.
And then we have a question on how can someone get more involved in this kind of research. This person has a BS in watershed science and works in wastewater treatment down in Coos Bay.
Oh, great.
So are there ways that they could help contribute to your research regarding their water quality data?
Oh, that's so great. Yeah, so I would direct them probably to the folks at South Slough at the National Estuarine Research Reserve there in Charleston, and or folks at the Oregon Institute for Marine Biology, which is University of Oregon's marine lab in Charleston as well. And just kind of, you know, reach out to people about how you could be involved either in kind of a citizen science aspect. They do a lot of that already at South Slough and at OIMB. And then, you know, just offering if there's data that you have available that, you know, could be of interest to researchers, and just kind of starting those conversations about the types of information that they need or involvement that they need, and how you can contribute. And then also, you know, there's been a relatively strong movement just to get this long-term data collection going, and specifically related to pH and carbon dioxide in the water column, because we don't have a lot of historical data about that, and particularly in estuaries. And so there's folks, including Francis Chan, my advisor at Oregon State, who works on kind of distributing a network of sensors, and he calls it basically the Netflix model, which is really old school Netflix, where they sent you a DVD, you watch the DVD, you send it back to them, they send you a new one. And so he basically does that, but with pH sensors, and sends it to folks like people involved with Surfrider or other citizen science groups, sends them a sensor, they deploy it in their specified location. It collects data for several months at a time. Then you mail it back to him. He downloads the data, cleans it up, sends you a fresh sensor, and you deploy it again. And so it kind of reduces a lot of those logistical costs associated with just general environmental monitoring. And so I would encourage them, if they have a spot where they think it would be feasible to potentially deploy a sensor, that getting in touch with people at South Slough, or like I said, my advisor, Francis Chan, would be a great way to go.
Very cool. And that looks like that wraps the questions. So with that said, thank you everybody again for attending. And as I mentioned before, we will go ahead and post a replay of this on our YouTube channel. It's the Cape Perpetual Collaborative YouTube channel. Thank you all for coming, and have a wonderful week, and hopefully see you next month.
Thank you.
Thank you, Caitlin.
Absolutely.